What I'm going to be talking about today are not the microbes on our skin, actually, but environmental microbes, so those naturally found in household dust, and how those can interact with ambient moisture in the form of relative humidity in the air to contribute to the chemistry of our homes. And we're focusing specifically on volatile organic compounds, or VOCs, so those compounds that become airborne. And here I'm showing a pie chart of potential sources of VOCs in homes. And we're specifically focusing on the piece about environmental microbes, both in a qualitative and quantitative sense. So how big is that slice of the pie, and what are the specific contributions? Now, I'm presenting these sources as static when all of these are actually dynamic. So all of these have sort of background emission sources interrupted with periodic spikes or enhancements, as we heard about earlier. Environmental microbes are going to be no exception to that dynamic. And second thing to point out is many of VOCs that are microbial are not unique. So there's microbial and non-microbial sources for VOCs. And a big challenge in this work is disentangling the two and isolating the microbial source for many of these VOCs. So microbes contribute to the environmental quality of buildings, both as a direct source of chemicals. As part of their normal metabolic processes, they will emit chemical products, but also in terms of transforming chemicals in their environment. So for example, microbes can actually or play a role in the degradation of plasticizers in household dust. And these chemicals not only affect our perception of that space and that building, but it has important implications for our exposures. So the one factor that's preventing all of our homes or all of our buildings from looking like this is water. So how much water is available? And water availability water can be available in buildings for a variety of reasons. They can be from floods, or from a building failure, generic term for something like a plumbing leak. But it can also be from relative humidity, so water that's just in the air. And ultimately, what we're trying to understand is what these environmental microbes do with the water that is available to them. So our previous work in this area began on surfaces that were intended to be wet, so kitchen sinks and shower stalls. And on the left, we have a microscope image of one of these coupons that was left in a kitchen sink. And you can see these coupons get coated in a film. And when you zoom in on these films, this ribbon of film ends up is sort of revealed to be a ribbon of bacillus-shaped bacteria that has grown up on these stainless steel coupons. And we showed that there's a relationship between the total VOCs that are coming off of these coupons and the biomass of the microbes that are on those surfaces, suggesting a link between microbial production of VOCs in your home. But most of our homes are not designed to be wet. So what we wanted to do is move to areas that are not intentionally wet. And we wanted to build a framework for the relative humidity conditions under which microbes are active and contribute to volatile chemistry. And I'll talk about two aspects of that work that's ongoing. One is to identify the critical relative humidity levels that support microbial growth. So this is a pretty basic question, but one that has important implications for building recommendations and building design. And the second agenda is to use the time of wetness model to really try to understand and isolate microbial and non-microbial contributions to volatile chemistry. So this research is enabled by using a combination of innovative techniques in both chemistry and biology. So we're using an instrument that Alan mentioned that can detect these organic compounds, some in the parts per trillion range, in real time. So offering us really finely resolved data. And we can combine this with biological tools. So we can not only look at the composition and the species of taxa that are there and their biomass using DNA-based techniques, But we can also look at specific gene profiles that are being expressed through metatranscriptomics. So I'll give a brief overview of what we're doing with this work. Is we have two sample types that we're working with, carpets and drywalls. And we autoclave sterilize them, so we subject them to a high-pressure oven to try to eliminate any sort of background microbes that may be there. And then we subject them to treatment. In carpets, we embed them with natural household dust. We also embed it with dust that's been irradiated to sort of maintain the dust matrix but eliminate the microbes. And then we also have carpet only. And we're looking at dust from 3 regions of the US and from 2 different carpet types. For drywall, similar thing. We inoculate naturally in homes, compare that to drywall without that have not been embedded, and compare. And again, we're looking at different regions of the US and in different paint types. So first, to show what we're finding in terms of microbial growth. So what we're looking at is the amount of fungal biomass on these samples as a function of the relative humidity that they were left to incubate. And what you can see is that in carpet, you see a large flux of microbes that are fungi that are proliferating when the relative humidity increases from 75% to 80%. And in drywall, you don't see the same sort of growth until 95% relative humidity. Now, I should point out that we make all our comparisons with sterilized samples because we want to disentangle this microbial and non-microbial contribution. But it turns out that the techniques that we're using reduce but did not entirely eliminate all microbial activity. So I'm just showing as a comparison what the fungal biomass was on samples that were completely sterile. And so they were quite marginal compared to when microbes were there, but they were still present. So when microbes were there, at high relative humidity, you saw something like 30 million spores per milligram of dust, and our sterilized samples are closer to 20,000. So we're seeing a drop, but it's not completely eliminated. And this bore out when we actually looked at the material. So we, as I mentioned, we irradiated dust to try to eliminate the microbes that were present. And so we sent this to a company that kills things, usually on medical equipment, using gamma radiation. So this was our sort of sterile dust. And then after we started getting these findings about maybe our sterile samples were not so sterile, we looked at what would happen when we grow these on a plate with food. And the top is your normal household dust. You see quite a rich flora. But on the bottom, we're looking at irradiated dust. And you can see it's not completely sterile. So what this shows is that dust is offering a really protective matrix for these microbes to persist, even under extreme circumstances. So next, moving on to the time of wetness model. And this is the model, or the idea, that microbes, or that environmental conditions in homes are going to fluctuate between those that are favorable for growth and those that aren't. And this is specifically in the context, again, of water, of how much water is available to them. And so we cycle these samples through periods of high and low relative humidity, where high relative humidity is 85 and low is 50. And the relative humidity of 85 is quite high, but not completely unrealistic in carpets, where it can also be high. Carpet relative humidity is often higher than ambient air relative humidity, or for example, after a shower. So these are periods of high relative humidity, but realistic. And in each of these plots, the low relative humidity are in gray and the high relative humidity is high. And I want to point out sort of how or not these compounds respond to cycling relative humidity and how that may indicate a microbial or non-microbial source. So here we're looking at benzaldehyde, and on the left, the concentration of this compound is not responding to relative humidity. However, on painted drywall, it is. And you can see that the dark blue lines have microbial inoculum on them, and the light blue line does not. And so this this sort of difference between the substrate background and those with microbes we're attributing to coming, we hypothesize are coming from the microbes, actually being generated by the microorganisms themselves when they're growing and proliferating under high relative humidity. So another compound is a C8 unsaturated alcohol that's commonly called the mushroom alcohol because it's produced by a lot of fungi. And in both carpet and drywall, it drops, in the case of carpet, it drops to background when the relative humidity drops and responds, increases when the relative humidity increases. Same with painted drywall. And it's one thing to point out is that there's a lag in the response. So the concentration doesn't increase immediately when the relative humidity increases, which is entirely consistent with a biological origin for this compound because we know that from a quiescent state, microbes there's a little lag in how quickly they can sort of rebound and grow. So in addition to fungal markers or fungal VOCs, we also see indication of bacterial activity. So hydroxyphenyl acetate was a compound that was highly correlated with microbial biomass in our previous work. It's part of the styrene degradation pathway. We did not observe this in carpet, but again, we observed it under periods of high relative humidity in drywall. So this work is ongoing, but to sort of show where we would like to take this. So this dynamic processes of looking at how microbes and surfaces respond to varying relative humidity is really powerful as a way to disentangle the microbial and non-microbial source for many of these VOCs. And the next step is to link specific gene compositions and gene profiles with specific VOCs. And I think this is going to be powerful because it's going to allow us to not only isolate particular bacteria or sort of biological origins that are related to chemicals of interest, but maybe also flag some chemicals that are tracers of biological activity indoors. And I mean, I'll leave with the point that we've known for thousands of years that living in a damp and moldy building is bad for your health, and we still don't know why. And it's a really challenging problem. I don't want to minimize that, but I think these tools offer a really powerful approach forward to tackling that long-standing problem. So I'll thank the Sloan Foundation for funding this collaboration, which is the group I'll show here and there.